Yeah, thank you everybody for joining the live session on the topic here, which is really, it's spatial biology focused, but it's kind of an interesting time in science in that there's quite a bit of discovery that's gone on in spatial, but really trying to how can we bridge that discovery to translational gap in spatial biology. And we have with us today Zhihong Chen, who's the Chief Technical Officer of OCCAM Immune, who's kind of doing this work. Z, why don't you go ahead and introduce yourself briefly?
Sure. My name is Z. I'll just go by Z, which is the letter, happen to be the last in the alphabet. So I'm an assistant professor at the Department of Immunology and Immunotherapy here at the Icahn School of Medicine and Mount Sinai Hospital. Um, I'm also serve as CTO of OCCAM Immune. So my research interest has always been focusing on neuroinflammation, particularly those related to the innate immunity, how they interact or react and shape the disease microenvironment in central nervous system. So during that process, I also developed a strong passion for technology development and their implementation. So looking back, I'm among the first to use this 3D electron microscopy to look at the interactions between microglia and neurons. I'm also among the first to use multiphoton confocal microscopy to peek directly into the brain of living mouse to observe microglia and macrophages in brain tumors. So all those technologies are really transformative for my studies. So here today, I'm also very excited about this Orion, another ground-shattering technology, I would say, to share with everybody our experience in adapting this technology. And hopefully we can democratize it to everybody who's interested in using it.
Yeah, thanks, Z. And
Yeah,
also in the kind of spirit of interactivity, we
yeah.
definitely feel free. Anyone that's on there, participate, interrupt with with any questions that you have. There is a sort of a Q&A function on the Zoom bar. Just use that to ask questions. What I'll do. I'll give a few slides on the Orion technology and how we feel like it's transformational for translational spatial. I can't believe I said that. Um, but feel free to interrupt us with questions. Z will interrupt me with questions when Z's talking about what he's doing with our technology at Alchemy, I'll interrupt him. But just feel free to participate. Um, so yeah, so let me go ahead and give you kind of a general introduction, to what we think of as translational spatial at Raresight, and a little bit about what was behind the Orion system. So, the term spatial biology is used, a lot, but I think it means different things to different people. But I think the highest level is spatial biology is essentially evaluating tissues and in particular, microenvironments within tissues, which means you're using imaging technology, but you're trying to determine like what's going on and how that impacts human health. So it spans, very research oriented activities, kind of, in the last 10 years there's been an explosion of whole transcriptome level spatial information that's generated lots of insights in the research side. But it also includes clinically impactful single protein IHC tests and even really, H&E, right? So, although I think most pathologists doing that type of work don't necessarily consider themselves doing spatial biology, but they really are, right? And there's kind of a moment in time now where this is exciting, but it's also, yeah, I would say prior to the Orion introduction, there's been no practical way sort of to translate this tremendous amount of research insights into spatial biomarkers with actual clinical value. There's kind of this gulf. Um, and I won't get into kind of why we developed the system the way we did, but suffice it to say that this informed the Orion. which you can think of as a very high-plex single-round approach to spatial. And if we look at the translational continuum, it kind of goes from research to clinical. In the research side, you're looking to figure out, potentially valuable biomarkers in pilot studies that ideally you would want to validate as part of a larger scale clinical study. So you're looking for new biomarkers in this space. On the more clinical side, you're simply trying to reveal biomarkers with actual value, but they're typically done in large scale studies tied to clinical outcomes. So there's different sort of ways that or technologies that you would use in these different settings. And a lot of them are kind of described here. So in biomarker space on the protein side, you're typically downstream of high-plex, transcriptomics. And you're typically looking for, I don't know, 15 to 50 biomarkers to try to see which ones are useful. The study size is typically relatively small. Data quality is important, of course, for everybody, but it becomes more and more important as you go towards the clinical. We'll talk about that in a second. And of course, there's a run cost sensitivity. Certainly, if you have the technology at a CRO or at a core facility, you need to make sure that people can afford to run the experiments, right? So in the research side, there's lots of plex, there's lots of reagents per sample. In the clinical side, the throughput goes way up because typically you're building studies here tied to clinical outcomes and you're looking for Kaplan-Meier curve level statistics. So I would say normally people are doing at least 100 samples, right, Z, in order to
Yeah.
do that. So in that case, you also need affordable reagents not because the plex is high. The plex typically goes down. Typically, you already kind of know what cell types you're looking for, say immune response to therapy. You're not looking for novel biomarkers. You're looking for response to therapy, or maybe if you have retrospective cohorts, you might want to design a targeted study to sort of address what biomarkers might have positive impacts for outcomes. So those are large studies and need lots of reagents. The other thing that really happens that I've alluded to before, once you get into this sort of clinical space, you really want reliable results, right? So you need things like accuracy, reproducibility, single round tissue integrity, whole slide analysis, validated reagents, single lot studies, all these kind of things that typically people are talking about when they're talking about IHC, for example. But of course, IHC is limited to one marker typically per section. And that's not going to be quite sufficient, right, to resolve microenvironments, right, where there's different cell types and states, et cetera. So that clinical workflow is really what drove Orion because we needed some single round throughput and data quality at a plex, really that really wasn't available in the marketplace. We do address the high-plex research market. Obviously, cycle with the Orion. It's a 20-channel system. So if you need to do more than 20, you can certainly cycle and address that kind of high-plex activities. So Orion basically covers that sort of translational space on a single platform. 20-channel system, super fast, low run cost, tissue preserved, whole specimen, flexible panel design. I think Z, you'll probably allude to I don't know how many panels you've done at the OCCAM, but you've got quite a few. And that essentially translates into more data for publication. But on the translational side, really it's oversimplifying things, but Kaplan-Meier curves for clinical studies. Any tissue, any indication, I don't know, internally we've done 250 panels, every single tissue type. If you can, make them fluorescent, the scanner will see it. A simple example, there's also when you're in sort of spatial biology, of course, you're looking for quantitative outputs, which typically are called spatial biomarkers. I would say in our services group, and I don't know what you guys do at OCCAM, but I would say 95% of what people are asking in these clinical studies is what's the density cell types, in and around the tumor, for example, or it's cancer
Yeah.
or whatever. I mean, there's lots of spatial biomarkers. I don't know if there's any other ones that you typically analyze there at Occam. Does that
Yeah.
sound like what you're dealing with, would you say?
Yeah, I think that's one of the perspectives, but probably more questions can be asked from the same essay as well.
Yeah,
Density is one of the questions, or also distribution,
yeah,
neighborhood, and
right.
states of cells, all that.
Right. So yeah, basically what's going on inside, who's in the neighborhood, maybe how far they are
Yeah,
from your boundary.
yeah,
So I think, a lot of the computational papers have lots of fancy words around it, but that's
right,
basically what you're doing. You want,
right.
and it's kind of a nice interplay. It's a collaborative kind of analysis because a lot of times
Mhm.
you have pathologists involved that can look at the H&E and say that's kind of where the tumor is. Maybe you need a more, math-oriented computational person that's good at classifying cells. And if you have those two things, like chocolate and peanut butter, you can get your spatial biomarkers with that, right?
Yeah,
So,
yeah.
this is a simple example actually from a genus.
Hmm. But I
They,
have a question, Ted, here. So for
yep.
most of your services, do they come in just with established panels, or they also ask for customizations of certain markers?
It's a bit of both. Like this
Uh-huh.
particular one here from a genus, this was a pretty much off-the-shelf IO
Yeah.
biased panel. I would say usually people want to customize a little bit,
Hmm.
right?
Yeah,
You
that's our
know,
experience as well.
that's your experience as well. And
Hmm.
and
Hmm.
I think customization comes in two forms. Uh, One is just, like as you know, Z, we have over 100 biomarkers that we sell off the shelf reagents that are
Yeah.
already validated for the system. So, one simple, and we also have off the shelf panels. So,
Hmm.
non-custom is you just use one of the off the shelf panels. That's rarity. I would say that most
Yeah,
people would
right.
say, "Oh, I like these IO markers, but maybe I want to
Yeah.
add, TIGIT or something, for
Yeah.
example, right?"
Yeah.
So I would say most people will customize, probably over half of them within the library of reagents that we sell.
Hmm.
But then, of course, and you I'm sure experience this too, there are biomarkers that are very special to the particular investigator that's not part of the
Yeah.
catalog. So they'll want to create, reagents and add those. Either they can pay us to do it, you probably know how to do it, we train people how
Sure.
to do it, or the individual lab can do it. Um, it's pretty easy to do, right? In terms of
Yeah.
labeling, it's the conjugation to the argofluors that are used in the system. It's amine conjugation chemistry. I think the biggest amount of work is just validating the clone first in IHC,
Yeah.
right? Which is the
Yeah.
bulk of the work. But once that's done, the labeling's pretty simple. But yeah, I would
Yeah.
say, you know, 90% of the time there's some level of customization. Is that what you've
Sure.
seen? Yeah,
We see 100% actually.
100%. Yeah, that's not
Right.
surprising.
Right. Yeah. We'll touch upon that.
Yeah. I think that's the thing. There's way more proteins than
Yeah.
our
And also
channels, right?
speaks to the flexibility of the platform, right? You
Yeah,
have these backbone panels that cover most of the immune cells, immune
yeah,
markers. But what if you're studying non-immune disease, non-immune related questions that
exactly.
then you have the complexity,
Yeah,
right? The
exactly.
biggest panel we made so far is really from ground up. I think we only have two markers we got from our site and all the
Wow.
rest of
So
17.
two and then plus like 12 or 15 customs, right?
Yeah,
Yeah.
exactly.
Yeah.
Yeah.
Yeah, and you're touching on that point, right? Our catalog, I would say right now
Hmm.
is primarily oncology and autoimmunity biased,
Right,
mostly
right.
because our customer base, that's what they're doing. There's definitely foraying pretty
Yeah,
strongly into the neuro space right now. So
yeah,
you may not
yeah.
even be aware of that, Z, but
Right.
that part is growing. And then certainly a lot of people doing structural stuff like basement membrane or whatever.
Hmm.
I've seen
Hmm.
several customers that have sort of ground up some of those
Yeah,
markers where not necessarily even cellular markers they're looking at. They're more looking at sort of architectural
sure.
markers, right? But basically anything you can validate by IHC, you're going to be able to see it by the
Yeah,
Orion, right?
yeah.
So
That's our experience as well.
focused on making sure that that was a reality for the users. Yeah, so you have all this sort of complexity, right, in terms of choices of biomarkers or whatever, building it there, that, you will then stain your sample, image it, but then how do you turn these images and biomarkers into a spatial biomarker, right? So in this case, a genus had empirically found that if they gave dual checkpoint inhibitor immunotherapy to patients, they had colorectal cancer. In a subset of those patients, they had dramatic responses, meaning that the tumor, had shrunk dramatically. So what they're really interested in is like, well, what types of cell types were being recruited into the tumor space as a result of treatment. So they enrolled as a prospective trial. They enrolled 12 patients, got pre-treatment corneal biopsies and post-treatment resections. You can see the corneal biopsy here, resection there. This is the pathologist-drawn region of interest where the tumor is. And essentially, here, this is one of the resections, looked like normal colonic mucosa. What's not normal, these T cells white here, there's a lot of T cells. And if you pan to the right, that's where the active polyp is. You can see at least T cell invasion. So the images are great to show, but you want to really quantify that. So again,
Yeah.
We essentially quantified the density of B cells, macrophages, T cells, et cetera, in the biopsy versus the post-treatment resection. It saw a major recruitment of all the major cell types as a result of therapy, right? So that's a very simple example of the types of outputs people are typically looking for in the more clinically oriented, spatial biology programs. You can certainly do retrospective studies. So, this is a study in Harvard Nature Cancer where they had also colorectal, but they had this massive, cohort of blocks actually where they had clinical outcome data. And they used a 17-plex panel really to try to find lower dimensional combinations that gave good Kaplan-Meier curves. So that's kind of they were using it more to propose possible prognostic test, right? Again, needing that sort of throughput, that combination of, data, spatial context, plex, and throughput, right? So, yeah, with that, any other questions you have on Z before I kind of turn it over to what you're doing? You ready?
We're good.
All right. So let me escape out of this slideshow and then you can share yours. Go ahead.
Sure. Okay, thank you so much, Ted, and thanks for this opportunity. So I had this deck up, but feel free to interrupt me with questions or from yourself or from the audience. So a quick introduction on OCCAM. OCCAM Immune is an academic research organization, or ARO. So we offer a comprehensive high-throughput immunoassays to our biopharma partners or industry partners. So the ARO, you probably don't hear it often, but it's not dissimilar to a CRO. But what makes us unique is our academic DNA. So we're deeply rooted in academia, in Mount Sinai Hospital, the Icahn School of Medicine here in New York City. So Mount Sinai may not be a household name, but it's really a small kind of boutique hospital located in the Upper East Side of Manhattan, the center of New York City, but it really punches far above its weight. So Mount Sinai has been consistently recognized one of the best hospitals in the world by U.S. News and World Reports. And just the last fiscal year, we're ranked the 11th in the nation in terms of NIH funding. So that really speaks to our power, a powerhouse of innovation. That's really where the new diagnostics or therapies happen. So all that, consider that with not really a backing of a comprehensive university. So it is under this backdrop that we launched OCCAM Immune, combining academic excellence with the industry-focused mindset. And so although the OCCAM was just launched last January, but it's really built upon a long-standing, really legendary, if you will, a research center, which we call it Human Immune Monitoring Center, which we'll come back and talk a little bit more about. Now, our vision is to become one-stop partner to provide this comprehensive immune assays across all possible disease, right? Actually, not just immune-related diseases, but now expand to other diseases, including cardiovascular and neurodegenerative, all those diseases. We achieve this by combining cutting-edge technologies together with our deep expertise in experiment design, execution, as well as data interpretation. So we're also merging academic rigor plus industry efficiency. So come to our mission, we really aim to translate all this high quality data, clinical data, into clear, actionable insights, be it driving the biomarker discoveries or validations, or unveiling the mode of actions of the assets that's under investigation. So we actually really started from a really humble root. When we first established the Human Immune Monitoring Center, HMC, that was about over 15-ish years ago. We just started with 2 technicians and one simple flow cytometer, mostly process clinical samples in a simple way. But over the years, we saw a quite exponential growth under the leadership of Dr. Miriam Marat, a prominent immunologist, as well as Dr. Zhihong Chen. I think both are pretty incredible visionaries. So it's under their leadership we see growth, and also along the ways we picked up all these technologies that are cutting edge of their times. And for CyTOF, now so far we still have the most set of machines in any institution in the country. And we also were the first to introduce Olink PEA assays here, as well as large productions of single-cell sequencing and spectral flow cytometry. So because of this success, we established OCCAM just last year. So the aim is really to provide our pharma partners with all these amazing infrastructures that we already built and allow them to easily access and leverage all these cutting-edge technologies to accelerate their discovery and also translate science into better therapeutics with speed. So some statistics just to illustrate our capacity and our excellence here. So we've been managing more than 60 clinical samples every single year, clinical trials, clinical studies. And combined, we have accumulated more than 500,000 individual clinical samples in our custody, in our management. So this is kind of incomplete tally of the number of publications that we co-authored or authored over the years. So with some of the better known names of the journals listed here. So really come talk to us and work with us, see how our technologies, our service can elevate your research to a new level. So these are the assays we provide. So our workflow really starts, it takes a really streamlined and end-to-end approach from sample management all the way to data interpretation. So for samples, we start with the processing under our robust standard SOPs and manage them or track them with our laboratory information manager or LIMS system. And for tissue assays, we have multiplexed F Orion, which is focus of today. And we also have this spatial transcriptomics platforms from 10x, both VZM HD and Xenium platforms. And for cellular assays, as mentioned earlier, we have this mass cytometry CyTOF and plus spectral flow cytometries, which are also capable of high parameters, like 40 markers at a time. So really niche to us is also have these functional assays, including ex vivo antigen-specific T cells and basophil activation assays. And on the soluble factor side, we have this O-link assay, which is very powerful proteomic assay, which can simultaneously look at more than 5,000 soluble proteins in one drop of blood. That we also have this more classical low-plex assays that is well established and well used. And molecular assays, we have single cell sequence, as I mentioned, different varieties, different flavors of single cell RNA, ATAC, TCR, BCR, using the 10-axis suite of Chromium assays. And we also have BD Rhapsody, the microplate-based assay as well. And on the microbiome side, we do 16S RNA amplification. So sequencing. So I think at this point, Dr. Eric Bombley, our CEO, always like to say all these assays generate a tsunami of data. That's his words, and this is a fact. So, but fortunately, we have this group of dedicated and very talented bioinformaticians who are very well versed to analyze this type of data. So again,
Yeah,
Orion.
there's a question from
Sure,
Z.
come.
Because you obviously have an impressive array of tools and experience and personnel, right?
Yeah.
And the subset on tissue analysis, right? In that sort of spatial space,
Yeah.
you're kind of alluding to data analysis, interpretation, et cetera. So, the question's mostly focused on, when you're kind of going towards the spatial space, what are the top challenges you face? Right?
You're right.
Because yeah,
You're right on spot, Ted.
yeah,
Right.
right,
Yeah.
yeah,
So I think the wide bench workflow is quite mature now to this point. We haven't really encountered any difficulties in that.
right.
I
So
think the
staining is
most
okay.
challenging part,
Yeah,
yeah, is data analysis is the most challenging part.
right. Okay.
So yeah, so there are platforms out there, softwares out there that is commercially available. So a couple of things, and I don't. So one thing is that many of them are kind of the commercial side is kind of quite pricey,
Mhm.
right? And so us as well as one of those kind of groups that's capable to make their own. So we have these implementations to establish our own pipelines of
Okay.
analysis,
Right.
taking modules or toolkits from various available resources.
Right.
But again, the challenge is there, cell segmentation and cell annotation. I think those are actually critical in terms of downstream analysis.
Mm.
But I can see there's a ton of work that needs to be done to
Yeah.
improve.
Yeah. Okay. Yeah, makes sense. There's another
Yeah,
question. Yeah,
sure.
because related again, you have essentially the transcriptomic stuff and then
Yeah,
IF. Like, do people choose one or the other, or they try to integrate them together, or are they different types of clients that are going after, the
right.
RNA versus the IF, or what's been your experience so far there?
Right. That's also a great question. So
Yeah.
I think they addressed different things. As you alluded earlier, there's kind of two stream, if you will. There's this more discovery focused essays and also validation or translational essays. I think they cover both sides. With the spatial transcriptome is more of discovery platform, and the multiplex F is more of validation or
Yeah.
translation platform. So I think we'll get the best of the both worlds to combine the two together, and you go with the high-plex. With the VZMHD, we can do the whole transcriptome. And with Xenium, you can do up to 5,000 genes targets now, which we just established. And on top of that, from what you discover from there and then translate to validation with the Orion with relatively smaller panel that's manageable. And that's, I think, the best of the two worlds.
Yeah, yeah, makes sense.
Yeah, right. Right. I think I would just ask this, almost the same question by about 10x executive just last Friday. So I think, right,
Popular
right,
question, yeah.
right. I think great minds thinks the same because everybody probably have the same vision.
Hmm.
Mhm.
I mean, do you find are they different groups of people or that like that some people they're interested just in their discovery and other people are more downstream, or there's some people that are
You
overlapping
know,
or
yeah, that's interesting question. I think from our observation, there's really a divide
I see.
between the two. It's more of our academia because we interface with both academia and the industry.
Yeah.
It seems that our academic clients is more going to the discovery side, and our
Right.
industry side, they came with a very straight, very clear goal, very clear question,
Yeah, it makes
right?
sense.
More of the on the data side. Okay, more questions?
No, so go ahead.
No, let's just move on here.
Yeah.
What's the, yeah, that's our focus on Orion. So of course, to maintain the cutting edge, we actually attempted this different platform. I don't know if we talked about this Ted before. So actually, years back, we acquired this system called MIBI, multiplexed ion beam imaging. So back then, it's really ambitious project, but the equipment or the technique is still in the beta stage. Um, so long story short, it didn't really deliver what it promised. So we will have to give it up, a multi-million dollar machinery, just really have to, well, stop using it. So and in between MIBI and Orion during the past 2 or 3 years, I think there's an explosion of technology development in this space. Now we see many novel platforms, but mostly focused on cyclic standings at their principle. And those are, I'd say, amazing platforms. But what really stand out for Orion, I think I just stumbled upon it from a YouTube video a couple years back, and I was really impressed with all the images that were shown in that video. So what I think what resonated with me at that moment is this algorithm that the Orion uses, the spatial extraction. And I think for us immunologists, that's what we use, right? For flow cytometry, we use for years, decades now, especially with the recent introduction of spectral flow cytometry. That's really close to our understanding. So make it easy for us to understand it, also make it easy for us to embrace the new technique.
Right. I guess what you're
So
describing too, because I know the MIBI quite well. I
yeah,
guess what's common about MIBI and Orion is they are that single round high-plex approach, right?
yeah, yeah,
It's just the MIBI and Hyperion have, kind of went away from spectral overlap by going to metals, right? So,
right, that's true.
you know, um, so I guess they're both similar in terms of flow cytometry staining protocols almost, right? Make a master
Yeah,
mix, stain everything at once.
right.
Um, but yeah, the Orion is fluorescence based, so therefore it needs to deal with spectral overlap,
This
whereas
package I'm mixing, right?
there's very little, mostly stemming, I guess, from impurities in the ions or whatever. But yeah, it's
That's true.
interesting that you guys see intrinsically were attracted to the single round high-plex approach. Is do you
Yeah,
why?
I think
So
right. I think a couple reasons. One
yeah.
thing is that I think this is a sweet spot with 15, 20 to 20-ish
Mm,
plexes, right? That covers most of your questions. And if
mm,
you have more, even too expanded panels, that the data could be well overwhelming. Another thing is that because OCCAM is industry facing, as you say, it's many of the customers coming with very clear question what they want to address, and we're defined size panels that we can perform.
mm,
So I think the platform fits us really well for our purpose.
right.
Right. So put this side by side, once bitten, twice shy, right? So be careful about this. So Dr. Kim again came to me and said, Z, why don't you just go to Seattle to test it out yourself with your own hands and bring some of our sections.
Yeah, I
So
remember
I
that.
went. You remember, so I went. Here we go. This is about, I think, exact almost 2 years back. when I was still young. And here you are, Ted, and you were there, I think, accompany my visit for the entire 2 or 3 days.
Mhm.
And thank you so much for your time and appreciate your hospitality. But at the same trip, I also met a lot of great colleagues here and maintained contact with them throughout the 2 years time. And Edward, Josh, and Danny. So what I don't have here is Melinda, Dr. Melinda Duplessis, our field application scientist, um, which whom I don't have picture
Yeah,
either.
she
But
was in the field
she,
training
right,
everybody. Yeah.
yeah, but she will always have place on my slide deck. She always has place close to my heart as well because our success really owed tremendous to her as well. So she was the one that held our hands step by step, help us establish the system. So up to this time, we still have this bi-weekly meeting between Melinda and Edward and our team here at OCCAM to discuss what we have encountered, what questions, problems we encountered in the past 2 weeks, and also troubleshooting with us. And they also listen to our feedbacks kind of patiently. So to that, I want to mention the level of customer service and customer support from Orion is really unprecedented. So we interact with so many companies, and I would say there's only probably a couple that can really have this vested customer support. And so because of that support, we can also provide the best quality essays to our customer as well. So I also have the opportunity to look behind the scenes. So this is the assembly in action of a Orion machine. So it's really a small kind of small footprint machine, and this is all peeled back. But a disclaimer here is that the machine we got is fully dressed and fully behaved. Um, you're right. It has been a workhorse for us for the past. We got it last summer, I think, past a little bit over a year. So we have built more than 20 panels, more than 2 dozen panels, with some of the examples highlight here. Colorectal cancer, as you shown earlier as well, multiple myeloma from the bone marrow, and hepatocellular liver tissues, non-small cell lung cancer, as well as small cell lung cancer of the neuroendocrine origin, ovarian cancers, as well. So this is what I want to mention a bit more because it's more close to heart for immunologists. This is tertiary lymphoid structure that we found in one of the lung cancers. And this is really a kind of disorganized cluster of immune cells together. It's kind of mimic a, lymph node, but it's not really a fully functional lymph node. But it's been found that the presence of TLS or tertiary lymphocytes, it can somehow used as a prognostic indicator for immune checkpoint therapies. So that is pretty interesting for us. So to your early questions, many times TLS is one thing to look for in our sections,
Yeah, great. We did get
right?
another question from the audience. It's
Sure.
probably for both of us. So that it's basically structured around, testing with the Orion because it's nice you've shown lots of panels there. This
Yeah.
question's more like, you know, any plans or attempts to transition to, CLIA for clinical testing, right?
Ah,
So,
it's a
uh,
great question.
yeah, so maybe you can start off there. I can also talk about, probably more of a question
Right.
for you, but you go
Right.
ahead. Yeah.
Yeah. I start off to say, Ted, how about a consideration of clear certification?
Okay, so you're gonna
Because
this
I
question.
remember this question that I asked when I was visiting, because I saw you have clear
Yeah.
certified equipment, but it's not, it's different, right? So I
Yeah.
think that that's a question we have all along, and that's I
Mhm.
think, a dream that it can come true.
Yeah, so definitely we do have other assays that are, CLIA compatible in the liquid
Hmm.
biopsy spaces.
Yeah.
And I do think the system's very, very amenable to LDT because, and the
Hmm.
main thing I would say, even over even the liquid biopsy stuff, we have CLIA tests in our lab for that.
Hmm.
I would say that what's really favorable in terms of administering this technology in the clinical setting is a kind of on the reagent side. We didn't talk a lot about that, but the reagents are anytime you want reproducible results and reliable outcome, which is pretty much what CLIA is demanding, right? That
Hmm.
you can rely on the result and you can get it over and over again, and right.
Hmm.
Uh, big part of it is the instrumentation, but for these, it's mostly the reagents, right? So,
Hmm.
um, and we thought a lot about that in terms of the approach we took to the
Hmm.
reagents that are used in the Orion system. And as you know,
Hmm.
these are antibodies directly conjugated to small molecule
Here
organic.
we go. I have a slide for you, Ted.
Excellent. Right. And you're probably right, validation on single channel versus IHC, et cetera. So, and the more complex the reagents are, and this actually led into the certain attributes of the instrument, because usually what you're doing with traditional microscopy is if you're trying to reliably identify low abundance biomarkers, you're going to need amplification.
Hmm.
Okay, if you have amplification, you have more complex reagents that might need to go on autostainers, and then the burden for reproducibility becomes higher just because of the complexity of staining even one biomarker. Now, I think the question gets at, of course, in the clinical setting, people are interested in 5 plus, 10 plex, how can they do this all in a reproducible manner?
Hmm.
So, you know, certainly with direct conjugates, it's very easy to validate those reagents. We also have 5 year shelf life. So many of the, you know, pharma partners that are working with us on multi-year studies, it's all in a single lot, so there's no lot bridges.
Hmm.
The staining is super reproducible because it's a simple master mix. It's applied in batches, so the staining is
Right.
done. Like when we're doing trials, we typically are imaging stuff from weeks ago while we're
Right.
building samples that are coming in over time. And once we get to like 20 or 24 samples, we'll stain them all in one batch,
Yeah.
right?
Yeah.
And it's a master mix, simple 2 hour stain, right? So all of these
Hmm.
attributes are very amenable to CLIA requirements.
Hmm. Hmm.
So I
But how about the equipment side?
equipment side, we do manufacture, you know, other devices under ISO. Um,
Sure.
um, and that is something that we certainly, are thinking heavily about with the Orion.
Hmm.
So yeah, I think that's the if there's any follow-up question to what I've said there in the audience, please ask it. But um, and we do have, like you are doing an ARO, we've sold into CROs already. We haven't done any ring trials like, you know, say 3 or 4 site trials. We certainly got internal data running with multiple operators on multiple instruments, the same assay, getting the same result.
Hmm. Hmm.
Um, but yeah, I think there's definitely a pathway to CLIA and certainly LDT with Orion for sure.
Okay. Right. That's great. Good to hear.
Yep. Yep.
So
But
good
excellent
segue
question.
on the reagent side. So as a list here, there's a lot of off-the-shelf antibodies that are already available. And if you're coming with a panel that is come from those, then you come in for a treat. So we can start your project right away. We can even probably finish your project within weeks, depend on the size of the study.
Yep.
Um, but as I said earlier, for the 20 panels that we built, there's none of them really come without any requirement for customization.
Yep.
So some examples here, they want to look at the TREM2 expression macrophages or plasma cells in multiple myeloma tissues with 138, or the brain tight junctions in brain vasculatures that we built from ground that I mentioned earlier, or even cancer markers that is unique to certain cancer types. So we can all customize to build a panel to suit your study. So some examples I put together here. This is one The first one is a collaboration that we did with Dr. Dolores Hambarzumian. Uh, Dr. Hambarzumian is really is a scientific director of the Brain Tumor Center at UPenn, and she's really a world-leading scientist in this space in brain tumor, particularly as related to the brain immune microenvironment, brain tumor immune microenvironment. So she came to us to ask for build a panel. So I just list some of these kind of markers on the top on macrophages, CD4, CD T cells, and along with some other markers, more of the standard markers, which I don't have space to show. But she also asked, how about this oligodendrocytes or tumor markers that we can add in as well, and as well as exercise, GFAP or ALDH. So we can all add in all that. And one unique marker that she asked is marker A. So because the data is not published, I can only use a pseudonym here. Um, so we asked why focus on marker A? I think this is one of the questions coming from the discovery phase. What they did is they used this preclinical genetic modified mouse model, look at the brain tumors. And for those who don't look at single cell data on a daily basis, what this splatter shows here is the cluster of cells, and they are grouped together by their transcriptome profiles. So when you look at the astrocytes right here in this corner, you know that they are highly expressing GFAP or LDH markers, which we can confirm and validate with our Orion panel. And they also found that in astrocytes, the population, they also have seed marker A. What they found in that study is that marker A is quite highly correlated to the patient's cognitive functions, neural functions, and overall well-being. So the question is whether this marker A can be used somehow reflect the differences in different subtypes of brain tumors. So we're working on it, and the manuscript is almost ready. So keep an eye out for the publication coming up pretty soon. A second example I have is the collaboration we had with the biotech company. So for most tumor-targeted therapies, is not surprising that cancer cells themselves are the target. The unique to this study is that this company actually not looking to directly kill the tumor cells, but a cell type in the microenvironment, which are considered to be the tumor accomplice. So this is the panel we built for them, and these are the cell types that we can identify from this panel. So this is a phase 1b trial, even though that's the primary outcome is not about efficacy, but we can already see these cell types that unique cell types can be reduced compared to baseline when I get the samples or tissue samples from on therapy patients. So one last example I have before I conclude is a collaboration we have with our very own Dr. Dmitry Zamarin here at Mount Sinai. And Dmitry is also a world-leading expert in gynecological oncology. And we know that immune checkpoint inhibition, IHC, been used extensively in the past years, really achieve unbelievable efficacies in some certain tumors, but not all. And particularly in this ovarian cancer, so they found some patients responded well, but not all. And so in this retrospective study, because the outcome is already known, so they stratified the patients into responders and non-responders, and we did Orion on them. So altogether, it's 40-ish subjects. So it's pretty extensive study. Now, what they found is that the outcome is really not correlated with how many cells, how many T cells, how many macrophages there. What is important is really their relative location. So really come back, Ted, to your question, what are they looking at? And probably this is unexpected, but really come to that this type of assay, this type of results really come only provided by Orion or spatial analysis.
I got
So I
another
think
question
that's
kind of related to that.
sure. Yeah,
Back to the analysis again.
yeah,
Because as you know, like these are beautiful images, but sort of converting them into quantitative
Numbers.
results is
Yeah.
important. So, how do you work with your clients to get what they need? I know that a lot of times we have people ask us to, and there's another question
Hmm.
actually coming about a panel design, which I'll ask in a second. But
Hmm.
a lot of times people, they'll pick a panel, they'll submit samples, they get run, you get these beautiful images, and then they start asking questions about the analysis. Do you find that, like, how do you work with your customers do they usually know exactly what to ask, or do you have to kind of guide them? Well, yeah, it's the same way.
Right.
A lot of times they're
I
like, yeah, do the quantitative. And you're like, well, what do you do?
right.
So
Right. I
do you
think
have a strategy to guide people, or what kind of how
yeah.
do you interact with people to get, the results that are useful to them, right?
Yeah, that's a great question. I think this is relatively new. That's relatively new to anybody, to everybody.
Yeah.
So there's really no set template that we can follow. But our process, the regular process is that we would discuss upfront for panel construction already. So it's more a focused panel design.
Hmm.
From there, we'll run once the image is generated, we'll run our standard pipeline. So you know all that. So you
So
just
just to stop you there, meaning at the
segment.
time of panel design, you're already talking about the
Yeah.
analysis output, right? It's so that
Yeah,
mark
you
because
will have
then
some idea.
I
So
see.
actually throughout this process, we work closely with our clients. There's many iterations back and forth. We provide some data and then they look at data because most times they are the expert of they are the domain expert of the certain diseases, right? And we provide more of data, so it's more of objective data. And
Yeah,
then what's the data sets, what the data leads you to, and they then come back to modify their questions, hypothesis. So I think through the iterations, many of them then will reach a conclusion and can reach interpretation of the data.
right. Okay, great.
Yeah.
Yeah. And I'm getting yeah, so I think we're coming to a close.
Yeah.
You know, and yeah, go ahead and conclude and then
Sure.
you know,
I just want to quickly conclude that this is we established OCCAM and we are very excited about this platform. So we want to share the excitement with the bigger community. So come talk to us if you're ready to run
yeah,
your samples.
yeah,
Yeah.
absolutely. Yeah,
Hmm.
definitely, contact, Occam, contact us with any questions you have about the technology. We can also, yeah, so this concludes the webinar, but just to let you know, if there are any other questions, we can stay on and discuss any other questions in the next few minutes. But yeah, other than that, yeah, great seeing you again, Z.
Of course.
It's super exciting to see what you're doing there with our technology and yeah, thanks
Yeah.
everyone.
Thank you so much for having me.